this stable isotope stuff works. So session objectives. By the end of the session, hopefully you'll be able to understand the difference between a, quote, normal metabolomics experiment and a flux experiment, or in other words, one involving stable isotopes. Hopefully you'll be able to understand how to perform an experiment using stable isotope tracer in your lab if you want to do that, and then know the potential applications of fluxomics or stable isotope resolved metabolomics, as Dr. Fan said, and also the output of the data, what it looks like and how to interpret it, at least to a first approximation. So here's an outline, all the slides that I'll talk about. We'll start with introductory material, some really basic stuff, which probably most of you are already familiar with, but just good to set a baseline. Give several examples of different types of studies you can do with fluxomics with stable isotopes, and then talk about some additional considerations that are important for these types of assays. Okay, so first, what is an isotope? I'm sure I don't really have to tell most of you what's going on with isotopes, but just a very convenient and useful part of the natural world that we have these things when you're a mass spectrometrist. So an isotope refers to a form of an atom or an element that has a different number of neutrons from the most common form of that element. So if we look at carbon, the most abundant form is carbon-12. It has an atomic number of 6 that defines it as carbon. That means it's got 6 protons. Its mass number of 12 means it's got 6 protons and 6 more neutrons that give it that mass. A stable isotope so carbon-13, which is what Dr. found a lot of her data were using. Tracers that contain carbon-13 has 6 protons, 7 neutrons. It is 1 Dalton higher in mass than carbon-12, and if you have a molecule with multiple carbon-13s, for each carbon-13 you add, you get 1 Dalton higher mass. The natural abundance, and we'll talk about this in a little bit, of that is about 1.1%. So of all the carbon atoms in your body, about 1.1% are carbon-13. Then to just go a little further, you can also have carbon-14. This is a radioactive isotope, and you're probably familiar with carbon-14. It's useful for tracer studies. Radiocarbon dating, we don't use this in metabolomics because we can't put radioactive stuff on the mass spec. And its abundance, its natural abundance is extremely low. It has to be synthesized by some sort of a nuclear technique to get it in any appreciable abundance. Stable isotopes, you can usually get them by enriching them from natural sources and then doing chemistry thereafter. Okay, so how are isotopes useful in metabolomics? As you've already gathered, low abundance natural isotopes can be enriched and used to form stable isotope or labeled molecules. So instead of conventional glucose off the shelf, which is unlabeled, has a molecular weight of 180 Daltons, if we substituted all of the carbon atoms in this molecule with 13C, then we would have uniformly labeled glucose, and its molecular weight would be 186 Daltons. Using this as a tracer would add extra mass to every downstream compound derived from this glucose, and you can use that to trace pathways. And that looks like something like this. You go from a mass isotopomer distribution or an isotope distribution that looks like this. You have some at the unlabeled mass and a little bit from the natural abundance of 13C in nature, and then if you add this glucose and it goes to form citric acid, you might form citric acid with 2 of the 6 carbons labeled, and so you see that as an increase in the signal at this mass 193. Okay, so now a term to get straight, which I think will become relevant in Garrison's talk next, and he'll probably tell you a little bit more about this. We have this question of isotopomers versus isotopologues. Not everyone in the stable isotope community uses these terms exactly the same way, but this is the accepted rigorous definition. So an isotopomer, so let's start an isotopologue. That's the simplest. So an isotopologue would refer to the same chemical compound. So in this case, let's talk about pyruvate, with a different number of label atoms. So there's 3 carbon atoms in here. If we're talking about labeling of pyruvate, you could either have 1, 2, or all 3 of those carbon atoms labeled as C13. But there's no specification of the position of the labeled atoms. So these 3 different isotopologues will have different masses. So you'd be able to measure those differences on a mass spectrometer easily enough. Isotopomers, on the other hand, refer to the same compound with the same number of labeled atoms, but a different position of those labeled atoms. So if we have 3 carbons, we got the 1, the 2, and the 3 carbons, we would have 3 different possible isotopomers of 13C1 pyruvate. You'd have 3 different possible isotopomers of 13C2 pyruvate, and only 1 isotopomer of the 13C3 or the 12C3. So that's just a term to keep straight as we talk about this. Okay, so with mass spec, so most of what we do here is mass spec. Can we tell? Can we identify isotopomers, not isotopologues, in LC-MS metabolomics, fluxomics data? Well, first let's talk about the questions we can answer. So, can we tell if something is labeled? Yes, we can tell if something's labeled. We can see a mass shift, easy enough. Can we tell how many carbons are labeled? Yes. We can look at the data and say, okay, we went from a distribution mainly in unlabeled to something where the most abundant, we still have a lot of the unlabeled, but we see the new label that's formed is mainly 13C2. So mostly 2 carbons have been labeled. Which of the carbons are labeled, on the other hand? Well, how are you going to get that information from this? You're not. With this data, we have no structural information. We have no idea which of the carbons were labeled. So in this LC-MS data, just acquired on a MS scanning instrument, all we get is the mass isotopologue information. We do not get isotopomer distribution. If you want that information, you can use other techniques. GCMS may help you because it may allow you to fragment the molecule and look at particular fragments that come. Maybe it would fragment your citric acid or your oleic acid into a component that has carbons 1, 2, and 3, and 4, 5, and 6 in separate fragments, and you could tell which ones were labeled. NMR can do an even better job with 13C NMR. You can get specific structural information, and oftentimes tell exactly which carbons are labeled. And that's useful because it gives you more techniques for tracing specific pathways. However, it makes data interpretation, it can make data interpretation that much more complex. Most of the data that we generate here is isotopologue information, and it gets you pretty far as interpreting relative flux. But you should know that is a limitation of LC-MS metabolomics data where we don't have a fragment and that. Okay, very good. So different tracers and systems. What can we use to measure flux? So there's lots of different tracers that you could use, but the most common atoms that are labeled and purchasable commercially, commonly used are carbon-13, 15 nitrogen, deuterium, and I should also add oxygen-18. So those are 4 commonly used tracer molecules. There are others out there that you might consider using, generally more expensive. You can get molecules that are fully labeled. In other words, all of the carbon atoms are replaced with 13C. You can get metabolites, molecules that are labeled only in specific positions. You could purchase glucose with only the 1 carbon labeled, or carbons 1 and 2 labeled. So there's a lot of options out there commercially available. If you get really fancy, you can get, if you're looking for a particular thing, you may be able to get synthesized for you the exact tracer with the exact labeling distribution that you want. Different systems that you can study. Very easy to do flux studies in bacteria and yeast. These are cells that turn over metabolites quickly. They will use very minimal media, and so you can put almost any universal substrate and look at flux and look at isotopomer distributions. You can also do mammalian cell cultures. So you can take a plate of cultured cells, add a tracer, as I'll talk to you about in a few minutes, and look at incorporation of stable isotope. You can do isolated mitochondria or isolated tissues. So Dr. Fan this morning talked about explants from tissues. That was something where the whole thing was done in vivo, and then you were just looking at labeling out. You may be able to isolate tissues from a living animal and culture them for a period of time and do a flux study that way. And you can also do studies in a whole animal or a human where the tracer is injected or given orally, and you can look for incorporation in the blood, in the metabolome in the blood, or in a tissue explant, et cetera. And so there's a lot of different potential pathways, a lot of different potential tracers you can use. We'll talk a little bit more about that in a minute. Okay, what information can you get from an experiment involving stable isotopomics, stable isotopes? So there's three basic classes of information that I wrote down here. There may be others that I didn't consider, but one thing, in what we might think would be the ideal information, would be to get an absolute measure of flux. In other words, a rate of turnover of a particular metabolite or associated with a particular metabolic pathway in an organism, in a cell, in a particular condition. And that would be something that could be quantified in terms of nanomole per minute, so an absolute rate of flux. Sounds good, very difficult to get. Confirm a pathway. So another thing, a simple thing you can do with a stable isotope tracer is am I synthesized? I have a metabolite. I want to know where it comes from. I think it's coming from this pathway, but it also might have this other possible route of synthesis. Tracers are a great way to confirm definitively what the origin of a particular metabolite is upstream and also downstream, where a metabolite comes from, where it goes. And although you'd think that these pathways are well defined, you can go online and look at these things that have been defined, many of them since the 1940s. In fact, you'll find that there's a lot that's yet unknown, a lot of things that don't follow the canonical arrows in a pathway, or connections that are not well established, or things that although competing pathways where you may find under one circumstance you have one pathway active and another under a different. So very useful for that. Relative flux is another somewhat easier thing to measure. Okay, and we'll talk more about that. That'll be the majority of what we spend our time on. So okay, how would you go about measuring absolute flux? Well, it turns out that the best way to do this is to achieve a steady state condition out of a predefined mixture of substrates that are labeled in a known way. And so what you want to do is wait a long period of time, achieve isotopic steady state, assess the isotopic steady state that you achieve under the different conditions that you are comparing, and then use a computational model to attempt to solve, based on the labeling patterns that you observe in your metabolites, and the networks that you feed into your model, to solve for the most probable rate of flux. And there's always going to be error associated with that. It's just an iterative fitting approach which tries to approximate the best fit for the isotope distribution that you observed. The problem here is that it's often impractical to wait to isotopic steady state. You may miss something important that would be apparent from this sort of transient state. Another thing is that computational modeling with any sort of accuracy is very, very challenging. It's only as good as the data you put in, and it's only as good as the metabolic network model that you feed it. So it turns out that although if you want to do this, really you need to find a lab where this is their research focus, and it's remained that way for years. And this is not how we focus on delivering data for most of our clients, although we have had core lab customers who are interested in doing this and will take data generated by the Metabolomics core and using their own resources and or other collaborations have attempted to do this sort of modeling. Okay, so this is out here on the percent enrichment. We're way out here on the later in the time axis. So to confirm a pathway or to measure relative flux, we can use a transient or dynamic condition. So instead of looking here at an isotopic steady state, we look at the change from unlabeled towards full labeling, and we want to see how quickly we go from here to here. So instead of looking way out here, we look at some earlier time point and compare two groups and say, okay, my control group has achieved this level of labeling. My treatment group at this time has achieved a lesser level of labeling. In general, that would imply that the rate of flux is higher in my control than in my treatment. There's certainly more complexity there, and better than just doing a single time point is to look at multiple time points and establish a time course. So then you not only look at a fixed time point and try to construe all your data from there. You see metabolites may turn over at different rates, and so you get a more complete picture by looking at a time course. Okay, so how to design a flux experiment? Well, first question to ask yourself is which pathways are of interest? Do I want to study everything I possibly can? Do I want to study central carbon metabolism? That's a common place to start within within flux analysis. Do I want to study glycolysis, TCA cycle, pentose phosphate pathway, associated metabolites? Am I interested in lipid synthesis? Am I interested in lipid metabolism? Am I interested in a particular unique pathway that is somewhere else? Cholesterol synthesis, who knows? There's lots of different places that you may want to start. And so that will inform your choice of tracer. And I'll talk a little bit more about that in a few minutes. You can choose a tracer that's very universal, that's a common fuel substrate for a variety of cells, and can go, eventually almost everywhere in metabolic pathways. Something like 13C6 glucose would be a good example. Or you could choose a specific tracer, something like a particular lipid that might get taken up by cells and metabolized and probe more directly your pathway of interest. Duration is another variable that you need to consider. You want to be long enough that you can see appreciable labeling. If you do your labeling for 10 seconds because you want to get the very earliest approximation, but you only achieve 0.05% labeling during that time, it won't be high enough of a signal for us to detect it compared to the background in our mass spec, and you won't have useful data. However, you don't want to go so long that all of the time points are at steady state, or you'll miss that dynamic change unless what you're trying to do is to model the data and you have a well-defined mixture of substrates, and that equilibrium is useful information to your outcome. So you want to be somewhere in the middle, ideally a time course of time points that let you look at that transition from unlabeled to labeled in a variety of different pathways or whatever you need to. Then you need to consider your conditions, particularly of the media. You want to make sure that the right tracer that you decided upon is in. You want to make sure that any undesired or interfering substrates are out. So if you just go buy DMEM off the shelf and then say, okay, I'm going to put my 13C glucose in there, the DMEM may contain glucose. Well, okay, maybe you're thinking and you buy DMEM without glucose. Well, the DMEM also may have glutamine and pyruvate in there. And so those are other substrates that cells like to metabolize. And so you may get a different picture from what you were expecting if you were trying to completely label your cells with glucose. So you want to select your media in such a way that you don't have interfering substrates unless that's what you want to have to study the competition. Okay, so the gist of all of that is it's impossible to define the right circumstance for every experiment in this type of a setting. You should contact the core before performing your experiment to help you design the study and purchase the right tracers and reagents. Definitely can't emphasize that enough for flux type studies. Okay, so just that said, how do you go? So I threw this slide in here because Chuck asked the question earlier on, and this does not portend to answer that question. So how do you select exactly the right tracer? Consider the pathway. As we already said, you want, if you're doing glycolysis, you might want a glucose-related tracer. How many carbons do you want to have labeled? 13C6 is a good, easy place to start. But if you want to start looking at tracing things specifically in downstream pathways like pentose phosphate pathway, you may be better off with a different form of labeling. Maybe 1,2 labeled glucose is a commonly used substrate. TCA cycle. Other substrates are also possible. Specific pathways, other tracers are possibilities. You just want to answer the question, will it be taken up by my cells? If it doesn't cross the cell membrane, it's useless as a metabolic tracer. Is it metabolized to an appreciable extent? If you're looking at something that is, maybe a valid pathway, but it only winds up being 0.1% of an intermediate that you want to study, you're going to have challenges. Will it induce other changes in the metabolome that you're not trying to study when you just want to be at stable at sort of a metabolic steady state, albeit not an isotopic steady state? So all those are things that you take into consideration. Here's a couple of references that make some mention of tracer selection and give some options. Some of them are more in the context of computational modeling and solving of flux patterns, but the information that they provide is still relevant. And so you can look at those and or other references to help you guide you, and really just consult with the core. Okay, so I'm going to move quickly through this example because you've seen enough of this in Dr. Phan's talk earlier this morning. Just to define what exactly, it probably helps to spell out how you might do a very basic time course in cultured cells using a simple tracer like 13C glucose. So you might start with several plates of unlabeled cultured cells. One hint here, I like to begin with, a lot of the times you use 6 12-well plates for cell culture because it's easier to handle. You don't have to take 1 million plates out of your incubator. For these studies, it generally, for metabolomics in general, for Flux especially, it often helps to be working with individual plates because you have to stagger your time course and quench cells at different times. And so if you're trying to quench one cell of a 6-well plate with liquid nitrogen, you're going to quench all of your cells and mess up metabolism. And so it helps to be working with individual plates, just a hint. Of course, it's a lot more of a pain to handle. So you can decide based on how you do your work. Exchange and unlabeled. So then the simplest thing to do is say, okay, I'm starting my Flux time course. I'm going to exchange my labeled media for a 13C tracer, like 13C glucose, and then you wait. First, you always want to take a sample with no labeling to sort of establish a baseline. Then maybe you wait for 30 minutes and 120 minutes or some additional time points. You rinse your cells, and Maureen mentioned yesterday, Anna mentioned yesterday, we have protocols you can look on our website for how you might do the rinse and quench procedure. I normally will recommend a rinse with ammonium acetate, or if you can't even do that, no rinse at all, and then quenching the cells by. Yes, I'm sorry.
We loaded the protocol into the folder, Mbox folder.
Great. So they're up there for you now. So find that file, take a look at it, let us know if you've got any questions. Thank you all. So we can go through that relatively quickly. And then you're going to quench your cells to capture metabolism, do extraction, et cetera. And then what happens? Okay, well, Teresa already showed you this in detail, but I'll click through because animations are fun. So you label your glucose. It goes into a TCA cycle. First thing you get in conventional Flux is mostly 2 carbons labeled. Then another turn of the TCA cycle, things get more complicated. Of course, you can have reverse directions, as Teresa's pointed out, much better than mine is right now. Okay, so we get that patterns of labeling in the data. What's that actually look like? Well, this is what it looks like until Garrison comes and helps us out with a better way. In the next talk, I'll tell you about that. But what we have is a lot of peaks, just like we would have in conventional metabolomics data, except instead of only looking at a single mass, we have to look at all of the possible masses for a given compound. So here we have aspartic acid, and we have all the different forms of labeled aspartic acid with the 3 carbons that could be labeled. And we can see, depending on our conditions, we might have only the natural isotope abundance. We don't get as far down as 13C4. We might have a lot of labeling down further. And so what we do in metabolomics, as I already told you yesterday, is we measure the peak area. That's our metric that is proportional to concentration. Hopefully, we do that for all of our metabolites, and then we look at the ratio of peak areas, and that gives you that ratio gives you the isotopic abundance that we look at to try to model Flux. And then going back and superimposing that on the TCA cycle, we can look at that isotope distribution for the various different pathways, and I've just put a few of them up here. You should be very familiar, again, from the talk this morning, so I don't need to spend a whole lot of time on that. Okay, so now into some more specific tricks that you may want to play with designing a Flux study. One thing that we can do, okay, so we talked about picking one tracer to study the pathways that you're interested in. Oftentimes, the best thing to do when you're dealing with a cell where you don't know how it behaves metabolically, the best thing as opposed to just picking 13C glucose and saying, I think I'll get enough information from that, you may want to use a group of different tracers. And so if the question is, what fuel do my cells utilize? What are they actively metabolizing? You may not want to limit yourself to 13C glucose. You may want to use other substrates. And a good combination or a good panel, this technique sort of was popularized in our lab by Mahmoud, so thanks to Mahmoud again, is to use 13C glucose, 13C glutamine, and 13C oleate, or another free fatty acid, and to create a media containing all 3 of these things, but only one of them labeled at a time, and then expose your cells to these 3 different media and observe the patterns of flux at similar time points using these different media compositions. And that way you have competition, right? There's the glutamine concentration is the same in all 3 of these media, so the cells have the option to choose it if they want to, if that's their preferred substrate, but you'll only see the flux that's coming from the tracer that's labeled in that study. And so This is just an example from some of Mahmoud's data, looking at some mouse and rat tissues. I believe these were macrophages, but not important in the context of this study. And so he was looking at glucose flux from this particular mouse or a group of mice. So you could see some labeling. You see it in 13C2 and 13C3 to a lesser extent. That's where you would expect a lot of your labeling to first show up from glucose. In glutamine, you see 13C4 labeling, meaning sort of conventional forward flux. So glutamine is in equilibrium with glutamate, which is in equilibrium with alpha-ketoglutarate, which can go through the TCA cycle and eventually form 4-carbon labeled citrate. And so that's where this is coming from. Oleate, so we see one important thing is you see a lot more labeling than you see glucose. So these particular cells are primed to metabolize free fatty acids at the given concentrations that we were looking at. I believe it was 100 micromolar oleic acid compared to, say, 10 millimolar glucose. They preferred to metabolize, and I apologize for using kind of silly terms, but they metabolized oleic acids to a great extent. And so compare this with these other rat macrophages. The rat samples metabolize glucose to a greater extent, glutamine to a much lesser extent, and oleate to an appreciable extent, but not quite as much as in the mice. So another thing to look at in this data is that the pattern of labeling is also important. So you might have noticed here, we not only just have this +4 labeling, we also have a little bit of +5. And I think Dr. Fan mentioned this earlier in her talks. The pattern of labeling can give you some information about what's going on. So as I mentioned, we go from, to generate 4-carbon labeled citrate from glutamine, we go in the forward direction. We go alpha-ketoglutamate, alpha-ketoglutarate forwards, and then 4 carbons get incorporated through oxaloacetate into citrate, which really should have 6 molecules. 2 of them just aren't labeled. But the 5-carbon citrate is kind of a backwards direction of flux. It's called reductive carboxylation, so formation of citrate from alpha-ketoglutarate in the reverse of the classic TCA cycle. And so this is something which is activated in cancer metabolism and also under various other metabolic conditions. And so this is a pathway of interest to many investigators. So look carefully at your pattern of metabolism depending on the tracer that you choose. Okay, so yeah, so that's what I just said. And yes, okay, so further you can look in where the oleate is going as well, whether it's whether pyruvate is forming acetyl-CoA or whether you're going through through another pathway to oxaloacetate from 3-carbon labeled. There's that example of where I wasn't prepared to discuss Mahmoud's data. Okay, so 2-carbon in vivo fatty acid uptake. So this is another example of Mahmoud's, which I think was pretty cool. So an industrial collaborator was interested in evaluating a drug for the treatment of hypertriglyceridemia and diabetes, and the question was, how does the drug affect fatty acid metabolism? So they devised an experiment where animals would be dosed with a control drug or the test drug of interest at a low or high dose or a vehicle. And the animal model was ZDF rats, which were an animal model of, in this case, hypertriglyceridemia. And then they did a fat tolerance test. And so the animals were given an oral gavage of corn oil containing our tracer, in this case, uniformly 13C labeled palmitate. And then plasma samples were collected every hour. So we were doing, this is an example of in vivo flux. And then they extracted lipids and analyzed them in various tissues and in the blood by LC-MS. Okay, and so just to give some background on fatty acid metabolism, oral fatty acid uptake so fatty acids are taken up by the intestine and form chylomicrons, which then enter the plasma, and then chylomicrons are eventually broken down in the blood to liberate the free fatty acids through the liver. And so it's a relatively complicated pathway that results in the production or in the transport of fatty acids to the blood. And I just butchered that pathway, so I apologize to those of you who are interested in that type of lipid biology. But Mahmoud looked at the intermediates in the blood at early time points, which are labeled triglycerides coming from an earlier stage, and then later on, which is coming from this breakdown of the chylomicron remnants in these particles later on. And so you can see different stages of flux. And so triglycerides, you can look for labeling. This is coming from labeled fatty acids. You'll see a shift of 16 Daltons indicative of incorporation of the labeled palmitate into the triglyceride species. And so you could see appreciable effects. So the drug so if you just look at the plasma concentrations, you can see it affects triglyceride abundance, but also it affects the rate of incorporation of the tracer into the plasma. So we see here in this study, the drug actually decreases the abundance of the labeled triglyceride in the plasma. So it's taking it out of the plasma, and it's effectively putting it into the adipose tissue. So it's encouraging triglyceride synthesis in adipose tissue from the free fatty acids that were taken in. I forget exactly what this time point is. I think it's after several hours. So you see that the drug has increased incorporation of 13C palmitate into the triglycerides in the adipose tissue, and that's how it's reducing the triglyceridemia, at least to a very first approximation. So you can get useful information out of these in vivo flux studies if you design your experiment appropriately. Okay, another type of experiment is a pulse chase, and so you may be familiar with pulse chase type experiments in the context of radioisotope tracers. Well, you can do them almost as well, or even better in some cases, with stable isotopes. So at times, a standard stable isotope labeling time course may not quite give you enough to know the origin of an altered metabolite profile. So say you saw glycerol 3-phosphate was decreased under a particular experimental condition, and you wanted to figure out why. Well, there's two possibilities. One could be, so glycerol 3-phosphate is derived from glycolysis and ultimately glucose. One possibility would be less glucose uptake, less synthesis. Another possibility would be more glucose consumption, and that flux through this pathway has increased, and so that the intermediates of glycolysis have decreased and in abundance, and that you're seeing lower concentrations reflective of increased flux. And so a way to design a pulse chase experiment, rather than just adding a C13 tracer and waiting, what we're going to do is we're going to add C13 glucose to a media, wait for a defined period of time for the metabolites to label up, and then remove that media and switch the media back for a defined period of time, usually a shorter period of time, with unlabeled glucose. And then you can effectively freeze the metabolism and no longer be building up any more labeling, and just look at the metabolites that have been formed. Where do they go? And so in this case, waited an additional 2 minutes, quenched metabolism, and looked at the outcome. And so the outcome of the data, we're going to look at a plot of area versus time. The 13C label, after 15 minutes, not surprisingly, you get, so we're just looking at 13C abundance is complete labeling. Glycolytic intermediates tend to get essentially fully labeled. And then when we do this step where we remove the media and add the media with 12C glucose, we go from unlabeled to mostly labeled, mostly unlabeled in one condition, but then in a comparison condition, we get a lesser degree of labeling. And so you can then look at those rates and say something about the rate of clearance and formation specifically, because we know how quickly the 13C went from this labeled form to these two unlabeled forms. And so that can be quantitated as a rate, and we can say that the clearance of glycerol 3-phosphate was higher under so these were, so this is unfortunately Mako didn't put in here this before. So these were serum and palmitate treated cells. And so palmitate treatment increased the rate of glycerol 3-phosphate clearance, but it even more dramatically increased the rate of glycerol 3-phosphate unlabeled formation. So we were able to tease apart these two components, whereas if we were just doing a standard time course, that might not be feasible. Okay, another application that I mentioned before is just confirming a pathway. So this is from the literature. I wanted to confirm that this was a valid pathway, that glutamate through N-carbamoyl aspartate ultimately resulted in synthesis of these nucleotide intermediates. Looking at this steady state abundance, we saw differences, but was this really the pathway that was being affected? And so they used a stable isotope tracer, glutamine, which they confirmed formed carbamoyl aspartate, and then looked downstream and saw that under their conditions of interest, that indeed an intermediate that was formed through this pathway was was this 15 nitrogen labeled UMP, and so that confirms that for this somewhat more obscure pathway that this was actually their metabolite intermediate of interest. And there's all sorts of other different applications that you can put on this. Okay, lots of other potential considerations for these sort of studies. Here's just a few. What is an appropriate control sample to use? So these are some mistakes that people can make in a stable isotope. So we've had people said before, okay, we want to treat all our cells the same way. We're going to give them all the stable isotope tracer, but for our unlabeled for our control sample, our time zero, we're just going to put the tracer on there quickly and take it off as quickly as possible because we don't want to treat our control cells differently from our experimental cells. They need to get the same number, the same exposure. We're just going to assume that the metabolism is not going to be fast enough, and we'll pull it off. So does that work? No, in fact, it does not. So even 10 seconds of exposure to labeled substrate, putting it on and taking it off, gives appreciable labeling in your control samples. So no labeling is the way to go. You want a nice clean separation between your labeled and your unlabeled samples, and so you think it's obvious, but it's happened more often, at least a few times in the history of the metabolomics course. So your control sample, no tracer. The other samples, tracers at appropriate time points. Okay, duration of the experiment. So the goal is to assess in this study example is to assess flux from glucose to glycerolipids. So in this case, there's two possibilities. So we're looking at now not central carbon metabolism. We're looking at lipid synthesis. And particularly, we're looking at this intermediate here, glycerol 3-phosphate, and that is to form a triglyceride. Glycerol 3-phosphate is an important intermediate. You condense the fatty acid with glycerol, and you form these glycerolipids. So one possibility for labeling of your glycerolipids is just labeling of your glycerol 3-phosphate. So this is a relatively short duration process. Synthesis of glycerol 3-phosphate happens on the order of minutes, seconds to minutes following introduction of glucose. And so within a short period of time, you can see a shift of diacylglycerol here. So this is a diglyceride. You can see a shift of the isotope distribution from the natural isotope to a +3 distribution, and that corresponds to incorporation of this 13C3 glycerol phosphate. But there's another possible way to label your glycerolipids, and that is, so we already mentioned this short one, that is the long duration. So if you put these cells in 13C glucose for 24 hours, now you get a different labeling distribution. You get a much larger mass shift. And so the long process is through the TCA cycle, you can do, and various downstream steps, you can do de novo fatty acid synthesis from glucose, and then these can get incorporated into glycerolipids. So if you're interested in looking at de novo fatty acid synthesis, you need to choose a much longer labeling time period because that's a much slower process. And so it took a day, overnight at a minimum, to see appreciable labeling. And so in 24 hours, you can see the labeling of palmitate that occurs, and you can look at different conditions and say, yes, this has affected my rate of fatty acid synthesis appreciably. Very good. Okay, so natural isotope abundance correction. This is another important caveat. So I told you at the beginning of the talk that 1.1% of all carbons are carbon-13. So there's no way to get rid of those in biological samples. That's going to be in your data. So when you look at data from an unlabeled raw chromatogram, you'll usually see for a small molecule the main mass as your lowest mass, the fully 12C species. But you'll see appreciable signal at a Dalton higher. And if it's a larger molecule, you may see up to 2-3 Daltons higher with appreciable amounts of signal at those masses. I shouldn't say labeling because this is just natural isotopic abundance. And so what we'd like to do, conceptually speaking, is to take all of this natural isotope data and sort of re-add it, if you will, condense it with the unlabeled data, because this is not labeling from a tracer. This is just what was there already. And so we would like to do some sort of a correction to account for that natural isotopic abundance. In data where you have an actual measurable amount of labeling, this results in a corrected signal, which is not that appreciably different. But when we have a flux study, then what happens is we avoid ambiguity. Was this signal at 13C2, some of it could have been coming from my natural isotopic abundance distribution, and some of it may not. Additionally, things that are 13C2 labeled will have a little bit of additional labeling corresponding to the natural isotope abundance in the other as yet unlabeled carbons, and we need to correct for that. So the fact that we have all of these multiple isotopes and multiple carbons means it's not just so simple as subtracting and adding. We need to use formulas that account for the relative distribution of the labeling, and really it winds up being a computational process. So there's various software out there that will do this process for you. The core lab has that software. So in general, when you submit samples to the core for flux analysis, we'll do natural isotopic abundance contribution for you. Garrison will talk about that's also a part of the software that he will present to you, and gives you a good way of doing that. So does it really matter? Does it make any difference in our interpretation? Well, yes, it does. So just looking at the natural isotopic abundance, so note this data, I've removed the 13C0 isotope, so these are only the labeled isotopes, if you will. In malate, we see a very subtle difference. We see an appreciable amount of 13C1, but not very much of anything else in the unlabeled sample, so this is time 0. I'm sorry for not putting those. This is 10 minutes, 30 minutes, and 120 minutes. And when you correct, you remove your unlabeled isotope, and you're looking only at the labels that came from your tracer. In a bigger molecule like NAD with 21 carbons, you get a very appreciable amount of natural isotopic abundance in many different isotopes. And so it's not imminently obvious necessarily which of these, which of your signal is coming from natural isotopic abundance and which is coming from your tracer. But after isotopic abundance correction, and there's noise in this data, so it's imperfect, but you can clearly see that the new labeling that's occurring is mainly in 13C5, which corresponds to incorporation of the ribose moiety into NAD. That's the first most rapidly turned over part of NAD synthesis. Okay, just to briefly mention computational modeling. So if you want to measure absolute fluxes, as I mentioned, you're going to need some sort of a computational model. Requires knowledge of all the relevant pathways, inputs and outputs. The input is the mass isotopomer or isotopologue distributions for all the metabolites you can detect, and then computational iterative fitting is used. The output is estimated rates of flux for all the reactions, but the limitations, it's only as good as your input data and metabolic network. And another limitation that it's very complex to get this right. So it's oftentimes best to start with a simpler study using transient labeling and looking at relative flux before you decide that this is really essential. Just a few other things. So remember, important to include an unlabeled control sample in your analysis. It helps put the flux data in context. Both peak area and percent label contain useful data, so you can look at the change in area with your labeling, and also just looking at everything normalized to 100% and looking at relative labeling. That's what this looks like in terms of raw data. Percent labeling is more sensitive and less subject to reproducibility areas. Both the pool size and the extent of labeling are important information. Sometimes it's useful to investigate the final or major products of a pathway rather than focusing on transient intermediates, because the complexity associated with the source and the sink fluxes. That said, sometimes there's information that you only get in the transient intermediates. And then generally, you don't want to take data from just a single metabolic intermediate and say, this is my measure of flux. If possible, you want to look for multiple metabolites in a pathway and build up a more comprehensive picture that way. Okay, so we've talked about a lot of assays. Just to sort of put it back in context, what's the workflow for a flux experiment? Well, it's basically the same as it is for any other metabolomics study, especially for flux studies. Please discuss with us before you get started. Perform the experiment, mostly you, sometimes with help. We do the extraction, we do the isotope correction, and we give you data, and then hopefully, working together, we can make sense out of your data. We'll talk, so Garrison is now going to talk to you about other software developments, a new software package called VistaFlux, which helps really deconvolute and automate what could be a very manual and laborious process. Makes our lives easier. Will make your life easier if you wind up doing a lot of this fluxomics in your own lab. And I think that's it. So with that, if we have time, can answer some questions.
what you explained is just for a targeted analysis or for untargeted
Great
analysis
question.
as well?
So I would recommend, if you're getting started in Fluxomics, you think of it primarily as a targeted workflow. It is much less complex to be trying, so it is possible, certainly, to put a tracer and perform untargeted metabolomics. You'll get data, but the challenge of untargeted metabolomics is bad enough in trying to identify unknown metabolites or features of interest that may not be identified with a metabolite name at the outset, without trying to add the complexity of a whole bunch of isotopes and a whole bunch of other tracers. Although there are occasions when that's what you need to do, I would recommend for most purposes begin with this as a targeted workflow. If you're going to do untargeted analysis, do that without a tracer. Although you can't study flux without from metabolite levels, oftentimes a change in flux will be reflected by some sort of a change in the metabolite level. So you may get a hint that something's going on with a particular pathway from your untargeted analysis, and then be able to go follow that up with tracers. That said, there are people who do untargeted fluxomics. It is possible. We can generate the data for you, but be very cautious when going to interpret that data because it's extremely complex.
In a typical flux experiment, would you completely exchange the carbon-12 to a tracer, or would you suggest a proportion?
A proportion, right. So sometimes yes, sometimes no. For a simple study, if you're going to follow the workflow where I showed you, and you're going to be doing transient labeling, and you're going to be looking for incorporation over a time course, for a first approach, I would recommend, in general, a complete exchange of media. There are some circumstances, if on the other hand, you're aiming for metabolic flux analysis with computational modeling, you probably don't want to entirely replace your media with fully labeled substrate or only a single substrate, because eventually, if you wait long enough, you're going to reach full labeling, and then you have no more information than what you had with the conventional metabolomics. There are occasions when you may want to do some, 50% labeling or something else. One occasion is when you don't want to subject your cells to a full media change, and you want to do something gentler, like spike in a 13C isotope and just watch what happens without a media change. So there are occasions when you may be dealing with a mixture of tracers. There may be occasions when you want to mix two tracers together, 13C glucose and glutamine in the same dish. Generally, I recommend first do a parallel labeling study with individual tracers before you start trying to interpret data that comes from putting multiple substrates in at once. But yeah, so hopefully that answers your question.
Yeah. Thank you.
Yeah. Right. So MSMS can sometimes give you a little bit of information about isotopic position. So if you get a fragment that has certain carbons on it, and you know what those carbons are, then you may be able to get a little bit of data about which carbons are labeled. What you can't usually, so LC-MSMS fragmentation, generally we'll get one or maybe two fragments from a given metabolite that are abundant enough to really do a good job quantitating. We won't get a unique fragment that has every single carbon in a molecule. So although we may be able to construe a little bit of information about which carbons are labeled, it's very complicated to, or it may just not be possible to find a unique fragment for each carbon to really define it. GCMS, the fragmentation is more extensive, so you stand a better chance, because of the nature of the ionization method, you stand a better chance of getting more unique fragments. It's still not perfect. You may not get a fragment that's unique to each molecule. But you're right. If you just need that answer of, is it in carbons 1 and 2, or is it in carbons 5 and 6, under some circumstances with some metabolites, we may be able to get that information from an LC-MS study. Okay. Mm-hmm. Right. Right, that's a good question. Electrospray ionization is a complicated process. I don't really pretend to understand it fully. Yes, you do have an enrichment of one charge in a particle, but I'm not probably the right physicist to tell you about exactly all of how it works. And we can talk more offline about that if you want. Other flux-related questions probably shouldn't. Okay. Yeah. Yeah, so that's a great thing, and I actually thought about incorporating those slides, or some slides that would suggest the useful information that you can get from both approaches. So Flux is really good at showing you information about particular pathways, or metabolic flux using a stable isotope tracer. What it's less good at doing is telling you total oxidative metabolism that's occurring. There's so many intermediates, so many things that you measure. It turns out that a nice way to do that is just to measure oxygen consumption and or CO2 production. And so that's exactly what the seahorse does. It will tell you how the total flux changes under different conditions. And a seahorse can tell you to some extent whether it's coming from oxidative metabolism or whether it's coming from glycolysis and from lactate production from anaerobic metabolism. And so, so by combining, that sort of global picture of what happens under different, conditions to my total rate of metabolism, and a look at the pathways saying which metabolic pathways specifically are showing alterations in flux, you can establish a much more complete picture. So really, they are very complementary. We do them all the time, and so it's a great approach. We do have a seahorse in the Metabolomics. The Michigan Nutrition Obesity Research Center. So you can talk with Chuck or with one of us about doing that if you're here at U of M, if you want to use that as a core service. So great question. Okay, probably should move on to the next speaker.
Thanks, Charles. All right, our next speaker is Garrison Birch